Hello everyone. So last we did with part 5 photosynthesis. We are done with the Calvin cycle. Now in the part 6 we will deal with the C4 pathway, which is also called as the Hatch and Slack pathway. Here C4 pathway is exhibited by the C4 plants which are found in a dry tropical region. C4 plants will show the C4 pathway as well as it will show the C3 pathway. We can see this in maize, sorghum. Now coming to the specialities of C4 plants. They will possess cranch anatomy. Cranch anatomy is a special type of leaf anatomy where it will be surrounded by large number of cells, where the vascular bundles will be surrounded by large number of cells called as bundle sheath cells. In the C3 plants, we were not introduced with bundle sheath cells. In the C3 plants, in the C3 cycle, we were introduced with the mesophyll cells. But C4 plants will possess bundle sheath cells in addition to mesophyll cells. And why there is a requirement of bundle sheath cell, we will come to know in the later part of this video. C4 plants can also tolerate high temperature. They can respond to high light intensities, greater productivity of biomass, and lacks photorespiration. Now, what is photorespiration? There is a topic in this chapter itself towards a later part. So there we will talk in detail about what is photorespiration. Basically, C4 plants do not exhibit photorespiration. That is what we need to keep in mind. So as I said, they have the mesophyll cells. So these C4 plants, in their mesophyll cells, they lack the Rubisco enzyme. What we saw in the C3 plants, that the mesophyll cells possess Rubisco enzyme, due to which the C3 pathway or the Calvin cycle took place. But if in the mesophyll cell they lack the Rubisco enzyme, then obviously the Calvin cycle will not be exhibited by the mesophyll cells of C4 plants. And instead of Rubisco enzyme, they are rich in the pappcase enzyme. So here in the C4 plants, the first primary CO2 acceptor is a 3-carbon molecule, phosphoenol pyruvate, known as PEP, and it is present in the mesophyll cells. And the enzyme responsible for this CO2 fixation is PAP carboxylase or pappcase. Here, the first stable CO2 product is a 4-carbon compound, 4-carbon organic acid, C4 acid, oxaloacetic acid, or we can also say it as OAA. As a result, in the mesophyll cell, production of oxaloacetic acid, C4 acid of 4-carbon compound takes place, and along with that it also forms other 4-carbon compound like malic acid, aspartic acid in the mesophyll cells itself. Now all of these acids will be transported to the bundle sheath cells. As I already mentioned that in the C4 plant there will be two types of cells, mesophyll cells, bundle sheath cells. Now we will come to know why there is addition of bundle sheath cells, right? Why do we need bundle sheath cells, or why do we discuss about bundle sheath cells? Actually, how to identify the C4 plants, it is by the presence of the bundle sheath cells. We can say that these are C4 plants. It's the characteristic feature we can say. So now, what is bundle sheath cell? It is found in C4 plants, as I have already mentioned. Large cells around the vascular bundles. It will possess large number of chloroplasts, thick walls impervious to gaseous exchange, and no intercellular spaces. There used to questions will come on this, sometimes statement based. It has less number of fluoroblasts, large number of fluoroblasts. So this way the statements might come, or which statements are regarding the bundle sheath cells if you have to identify. So if you know all of these points, then you will be able to easily tackle those kind of questions. Okay. And it will have no intercellular space. Sometimes we get confused whether it has intercellular space or not. So the answer is no, it doesn't have any intercellular space. So now all the acids that were produced in the mesophyll cells be it oxaloacetic acid, be it aspartic acid, be it malic acid, all are 4-carbon acids. All of these will be transferred to the bundle sheath cells. Now why there is addition of bundle sheath cells? Because it is rich in Rubisco enzyme. And if it is rich in Rubisco enzyme, it means it can take part in the Calvin cycle. It means Calvin cycle is possible. So bundle sheath cells exhibit the Calvin cycle. Okay. So in C4 plants also Calvin cycle takes place, and it takes place in the bundle sheath cells. And mesophyll cells, it is rich in pappcase enzyme, but bundle sheath cells lacks pappcase enzyme. And as I've already mentioned, bundle sheath cells, as they possess Rubisco, so they will exhibit the Calvin cycle. They will exhibit the C3 cycle. Now we have this diagram in the NCERT textbook. It is according to the new NCERT, so you need not worry. And here they are having two cells. One is mesophyll cell and the other is bundle sheath cell. In the mesophyll cell, as I said, the 3-carbon compound, okay, 3-carbon molecule. Primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate, which is known as PEP. So here the CO2 fixation will take place. One CO2 will come. Already it's a 3-carbon compound. CO2, if you add, it will become a 4-carbon compound, that is 4-carbon acid, C4 acid, oxaloacetic acid, and along with that malic acid, aspartic acid. All of these will be formed in the mesophyll cells. Okay. And then all of these acids will get transported to the bundle sheath cells. So the second one is bundle sheath cells. Okay. And now here what will happen is that this carbon 4 acid is going to get decomposed, it is going to dissociate into two compounds. One will be a 3-carbon compound and another is going to be CO2. This CO2 will go, or it will enter into the Calvin cycle, and that is why it is said that bundle sheath cell exhibits Calvin cycle. As I have already mentioned earlier, bundle sheath cell exhibits Calvin cycle. So basically here 4-carbon acid is going to divide into CO2 and a 3-carbon acid. This 3-carbon acid will be transported back to the mesophyll cells, where it will get regenerated as phosphoenol pyruvate, because in the Calvin cycle also previously we have seen that regeneration should take place, or else the cycle won't be continuously taking part. For the cycle to take place continuously, it's important that regeneration also takes place, and like everywhere, here also the third step is regeneration of the primary carbon acceptor. In Calvin cycle, the primary carbon acceptor was RuBP, a 5-carbon compound, so that used to get regenerated. Here primary carbon acceptor is phosphoenol pyruvate, PEP. So here regeneration of PEP or phosphoenol pyruvate is taking place. Okay. So as I said, carbon 3 acid will undergo this regeneration, and that CO2 that will be released will undergo Calvin cycle. Here the fixation will take place, and all that takes place in the Calvin cycle will take part. And that is why in the C4 plants we need bundle sheath cell, because mesophyll cell lack Rubisco enzyme, so it cannot undergo Calvin cycle. That is why in addition to that we have bundle sheath cell, which is rich in Rubisco enzyme, and it can undergo the Calvin cycle. So Calvin cycle is shown, or Calvin cycle, all the plants will undergo Calvin cycle. Okay, be it C3 plants, be it C4 plants, all will undergo Calvin cycle. Just the difference is C3 plants Calvin cycle will take place in the mesophyll cell, and in C4 plants the Calvin cycle takes place in bundle sheath cells, because mesophyll cells lack the Rubisco enzyme. I hope you have got it, why we need a bundle sheath cell. All the reasons have been disclosed. And regarding here, we can see that Calvin pathway is common to the C3 and C4 plants. Since both are going to exhibit, only in C4 it will be exhibited in the bundle sheath cell. Okay. The Calvin pathway occurs in all the mesophyll cells of the C3 plants. As I said, C4 plant, it does not take place in the mesophyll cells, but does so in the bundle sheath cells, because mesophyll cells will lack the Rubisco enzyme, but bundle sheath cells are rich in Rubisco enzyme, So here in C4 plants, in the bundle sheath cells, the Calvin cycle will take part. Here everything, if you read, you'll come to know as it is mentioned. Now the next topic we have with us is the Rubisco enzyme. Next topic we have is photorespiration. Now photorespiration, C3 plants will exhibit photorespiration and C4 plants doesn't exhibit photorespiration. Now the reason behind this is what we will come to know here, and most importantly, previously we came to know that C4 plants have a higher productivity. They have a higher productivity because they do not undergo photorespiration. Photorespiration is a wasteful process. Here, instead of production of ATP, utilization of ATP will take part to release carbon CO2. So it is a wasteful product because we want more and more ATP. We do not want utilization of ATP. So in photorespiration, it's a wasteful process, and C4 plants do not exhibit photorespiration, so they are more productive. C3 plants exhibit photorespiration, so they are less productive. Now coming to this part, this equation we have encountered prior in the Calvin cycle. The first step, CO2 fixation, I said RUBP is the primary carbon acceptor of 5 carbon compound, 1 CO2, both together 6 carbon. Instead we get a 2 molecule of a 3 carbon compound, PGA, in the presence of Rubisco enzyme. Here we need to know that Rubisco enzyme has 2 possibilities. It can bind with oxygen also, it can bind with carbon also. So Rubisco, ribulose bisphosphate carboxylase oxygenase. So it has 2 properties. In my prior lectures, I have mentioned that carboxylation will take part, but they mention oxygenation activity also in the Calvin cycle. But actually the reason was not there. The reason is here. Here it is there. That why? This reaction takes place when CO2 is abundant. But what if CO2 and O2 ratio becomes equal? Okay. Or if it increases its affinity to O2, then RUBP will bind with O2, then oxygenation will take place instead of carbon fixation. Then what will happen? That instead of forming 2 molecules of 3 PGA, there will be the formation of 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate. Phosphoglycerate is a 2 carbon compound, phosphoglycolate is also a 2 carbon compound. And as I have already mentioned earlier, that in this photorespiratory pathway, there will be no synthesis of ATP and NADPH. Instead, there is going to be utilization of CO2. Overall, what will happen? Instead utilization of ATP. Okay. So here there will be more CO2. Release of CO2 will take place by utilizing ATP. But in C4 plants, what happens? That CO2 concentration will always be more compared to O2. That is why there is no chance of them to undergo photorespiration. Because every time here 1 CO2 extra is produced, so that will always maintain the increased concentration of CO2, and as a result there is no chance for C4 plants to undergo photorespiration. But in C3 cycle, no extra CO2 is released. There is no equal concentration at one time. There will be equal concentration of CO2 and O2. In that case, oxygenation will take place and it will result into photorespiration, and photorespiration will decrease its productivity. That is why C3 plants productivity will decrease. And in C4 plants, as always, CO2 is released here, as CO2 concentration will be more here. So Rubisco will function as a carboxylase, minimizing the oxygenase activity. So that is why in C4 plants, always Rubisco is going to act as carboxylase and not as oxygenase. So that is the conclusion that photorespiration is a wasteful process, which is C3 plants will exhibit, C4 plants won't exhibit, so C4 plants are more productive. So we can quickly summarize C3 cycle and C4 cycle in 3 different points. We can differentiate it this way. So you can have a look, and this is how we can end up.